Scientists around the United States are pushing the ethical line by making embryos that are part human, part animal, in hopes of creating a better model to study disease in people and possibly even create transplant organs. These embryos are called chimeras. To create these chimeras, the scientists start with normal pig embryos and use gene editing techniques such as CRISPR-Cas9, which can target and change specific genes. In this case, the target is to remove the gene which creates the pancreas. CRISPR-Cas9 is a system that can recognize DNA sequences and modify or make changes to them. With this tool, scientists are able to locate and make changes to genes quicker and easier. The enzyme Cas9 follows a guide RNA sequence until it locates a desired DNA sequence and cuts the sequence. Here, it is a location where the gene of interest is, and from there, sequences may be added or removed. Once gene editing is completed, they inject human-induced pluripotent stem cells, which can turn into a cell or tissue in the body. In the embryos, the iPS are likely to take advantage of the missing gene for creating a pig pancreas and instead form a human pancreas. iPS cells can be formed from skin or blood cells, and in this embryonic pluripotent state, they become a source to create any type of human cell needed. The organs they create potentially form and would be able to match the patient and greatly reduce rejection of the new organ.
To get the chimeras to form and see results of the gene editing, the embryos need to be put into a living pig's uterus to develop for 28 days. This is done through surgical procedure to get to the uterus, and many embryos are injected with a special syringe. The embryos are only allowed to develop for 28 days because this is when organs and other primitive structures begin to form. This allows results of the test to be seen without risking too much controversy, since it is uncertain where the iPS cells will go. The goal at this point is to only grow a human pancreas, but if the cells change other organs of the embryo into human ones, such as a brain, it could create an animal with a consciousness like a man. This is unknown ground for researchers and scientists and raises questions about the morality of such experiments, so the embryos are not allowed to grow into a fully functional state. After the 28 incubation period, the embryos are then retrieved and dissected to see what the human stem cells are doing and if they are creating a pancreas and not some other tissues. Some of the concerns will be how will injecting human stem cells react to tissues? Is this on the lines of playing God? What happens if the chimeras are allowed to grow to adulthood? Some of the benefits are to better model to study disease in humans, ability to grow organs for people needing transplants. These transplants would have a greatly reduced risk of rejection.